but it might not be working. So who shall we see about that? How do we?
We can change the keys and you can use the other.
Hey, there's a light. There's a light now. Good. Yeah, so even if you can do that and you can watch the colony 300 times, of course you're still limited because you'd ultimately have to remember who all the other individuals were to know which one is which during interactions. Right? So there's been tons of problems associated with obtaining these types of data. And it's only really been in the last couple of years, 2 or 3 years, that with the advent of deep learning, it's been possible not only to track individuals within large collectives, such as these schooling fish, but also to be able to identify individuals with no markings whatsoever. So the animals need not be tagged. We can reconstruct, for example, the visual fields of individuals. These are termites. This was done in Brazil, actually, the termite study. These are desert locusts, rodents. Again, humans cannot see the difference between these animals. There's no way a human could see the difference between all of these fruit flies, but the software can. So these neural networks are very effective at training on these specialized tasks of identification and tracking. So it's really opened up a whole new range of possibilities. But nonetheless, theory has still been really important. And so today I'm going to sort of take you on a journey for how we began to study collective information exchange within groups. Because remember I told you yesterday, originally when we were studying the locusts, we thought that would be a great system for studying this type of collective cognition, collective intelligence. And then the discovery of cannibalism and these aggressive interactions, on one hand is super fascinating, but on the other makes the system not particularly appropriate for thinking about higher forms of intelligence emerging from interactions. But when we look at other groups, such as schooling fish or flocking birds, here we really do have a sense that the collective here, so this is what differentiates biological collectives from physical collectives like liquids, solids, and gas, plasma, is that here there's a real notion of information transfer. Natural selection, such as through predation, for millennia has exerted selection pressure on these individuals. So these are much better groups to think about for collective information processing. So neuroscientists, of course, focus on the individual brain as being the seat of intelligence. Here, what I'm going to discuss today is how connections among brains give rise to higher order forms of intelligence. And that's been much less well understood. And so this connection between cognition within the brain and cognition elsewhere, outside brains, across the animal kingdom is going to be the focus of today and tomorrow's first lecture. And if you're interested, I can send you these articles. I just was figuring out last night how I can send you stuff. But it's a relatively old review, but it's still the only one that I know of that really discusses these aspects of collective cognition in animals. So I'll forward you both of these two papers. They're pretty short. And so you can see why, with this multitude of interactions, that it's so difficult to tease apart how the system operates, how the system works. Yet we want to try to get inside the head of the individual to see how and why she behaves the way she does. Okay, so that's the real key challenge when studying collective behavior. So prior to being able to obtain these data, so we saw with the locusts, in around 2006, it was possible to get coarse data, not identifying individuals, not knowing if it's 2 or 3 individuals within or in a cluster. But if we go further back, it was just simply impossible to get the data at all. And so we started off with modeling these types of systems. And I'm going to show you pictures of fish because I have them from Matt McHenry, but we can think much more broadly. You can think about cell groups. You can think about flocking birds. Later on, especially tomorrow, I'll show you data from primates. So we can really think very broadly about what we mean here. And so we can try to characterize the types of interactions that we speculate are occurring via equations. And so individuals exist at centroid C, so that's the center of gravity of the individual. Here there's two individuals, individual I and individual J, that have these centroids, these positions at time t, and they also have velocities. They're moving through space. And broadly speaking, what we know is that a very high priority for these animals, especially for birds in flight, is to avoid collisions. So it's just like yourself, right? If something is about to collide with you, you will kind of focus on that and not on the other possible interactions you might exhibit. So this tends to be the highest priority behavior. And in some cases, as you saw with those schooling fish before, they're physically touching each other, or cells are compressing against each other. And so they can't infinitely compress. They can't infinitely go over one another. Now, in the V-shaped model, it did not have avoidance. You can have an infinite number of particles in one location in the V-shaped model. So even though there was this relationship with density, there was no capacity for the system to regulate density. Now we have the capacity for them to locally regulate, to not infinitely crowd into a point in space, which is an obviously biologically meaningful rule. However, when individuals get isolated from one another, we now assume that there's a tendency for them to become attracted towards each other. This is what gives groups the boundary, as was discussed yesterday. The V-shaped model, if you just let it run in normal space, they'd eventually noise would accumulate and they would just diffuse away from each other. There's nothing keeping the system together. There's no sort of surface tension to the system. So we maintain the V-shaped-like velocity orientation, tendency to match velocities with neighbors, but we've added the repulsion and we've added this longer range attraction to individuals. So this is the alignment term. So this was the only thing in the V-shaped model, and now it's just one of three components. So we've got repulsion, attraction, and alignment now. And one of the things that we can do is we can actually start thinking about what are the evolutionary drivers on these types of interaction rules. So I argued before that predation pressure is likely to have been a very important factor in the evolution of grouping. But it's very hard to test that. It's very hard to have animals exhibiting different types of social interaction rules, to put them out in the natural world, and then see which ones get predated more than others, which ultimately is what's really happening. So we had the idea that what we could do is we could have a virtual prey population, so they all look identical to each other, but they've got random rules initially in terms of their attraction, alignment, and avoidance behavior. But then we could have real predators hunting and exerting selection pressure on these virtual prey. So in practice, what we do is we put a thin film, a thin translucent film inside the tank. We project onto it these little dots. And these little dots are perceived to be prey items by the predators, like little zooplankton. And the little dots, they all look identical, but they have evolvable rules. And then we use hundreds of predators to exert selection pressure on the strategy space. And so we can see whether indeed predation pressure does cause individuals to group together. And so this is us looking through this thin film. Here are the little dots, and here's a predator coming towards the prey and targeting an individual. And they'll hover, and then they'll strike. So it's very clear. So you can see even when the mouth opens. So it's very clear which individual was targeted. And we can do all sorts of things like ask how long does it take to target prey and so on. And to summarize this, over evolutionary time, so we're sort of compressing thousands of years of evolution into weeks or months in the lab, we find very strong selection pressure. And the predators find it increasingly hard to hunt the prey. So what did the prey evolve to do? Well, the the number of attacks, so this is inversely proportional to the fitness, is indicated by red. So red means this behavioral strategy is heavily predated. White represents strategies that were least predated over evolutionary time. So selection basically selects for individuals to balance the attraction and the orientation. Alignment alone is not sufficient. Attraction alone, just forming groups that are relatively static, is not sufficient. The prey would readily evolve to balance both of these tendencies, even if they couldn't respond to the predator. We would like to do further experiments now in closed loop, where the prey can actually also respond, so there's a feedback. But here it's just the predator exerting selection pressure on the prey. So you can see that the selection pressure is very, very strong. And so this supports the view that in nature, this is one of the most important factors. Like when we look at these juvenile fish schools that we study, they get attacked. I can't remember the statistics, but it's probably once or twice a minute in some times of the day, in the mornings and in the evenings.
In the previous slide, we can see that the predator, it goes for the prey that's alone.
Yes, in that particular example, that prey, for example, exhibited no attraction and no orientation. So that's by far the most heavily predated
But
individual. They get selected out of the population really quick.
but for the predator, wouldn't be a better strategy to go for the group instead of going to the individual alone?
So it seems that in most of the systems we've studied, being alone is by far the most dangerous. But of course, and I'll come to why that is in a moment. But of course, being alone also means you're harder to find. So in certain environments, with certain shelters or turbidity, like how the light scatters in the water, it may pay to be sort of quiet and isolated and cryptic.
Mm-hmm.
But
I mean,
typically in these systems, it's a very high, the predation risk basically increases non-linearly. Actually, I do your side,
Yeah.
or single terms.
from the point of view of the prey, I understand it's better to be in a group because you share the danger of being predated. But for the predator, isn't a better strategy to go for the group because there are
I'll show you experimental data in the wild later that shows that the predators really struggle with big groups. They really struggle to get anyone from a big group. If they can slice the groups up into small units, small entities, their predation success shoots up.
okay.
So clear evidence that, and I'll explain why later on.
Okay,
why that's the case.
thank you.
Maybe those batteries weren't so new, eh? Yeah, this works. Yeah, sorry, were there further questions here? Yeah.
So this balance between orientation and attraction, I didn't comprehend it very well. Does one thing neglects the other? Like those things are opposites, so that they need to balance it?
No, I think what's happening is that if you think about it, if you have a very, very strong alignment, then the group is going to be very predictable in where it goes. If you have a very, very strong attraction, the group is going to be tightly cohesive but not really going to move. The groups that evolved here tended to have what's called protean behavior, where they'll move in one direction and then suddenly switch. And so we can't prove that from this particular study. But the predators do find it much harder to hunt these groups when they exhibit these sudden changes. So we think that's probably one of the features that's being selected for. But it's hard to prove that from the data we had.
And that switch of movement, that can only happen if they aren't all aligned. You need to have a...
Yeah, you need to balance it in this way to get those switches of movement.
Okay.
There's also, there's another factor which is related to the visual system of the predators, which is sort of visual confusion. And no one really knows why, but when you have multiple prey, even with humans, if you have multiple dots on a screen and you have to target one of them, it will take you longer and take more sort of brain power to target if there are others, if there are so-called distractors. And so it could be really a limitation of the vertebrate visual system. You know, it's just an inherent problem when you have multiple targets to choose among them. But I still wonder, like, how, you know, there's such strong selection to really focus on an individual, and this relates to other aspects. So if you make one of these individuals look different, it's screwed, right? It will be targeted, it will be very easily targeted. So in nature, we find that it's called the oddity effect. Any individual that looks or moves in a little bit of an odd way with respect to its neighbors gets targeted super easily. So it's as if the attention of the predator can now be drawn fully to that target. And so we tend to find groups, if you go and just collect fish schools, they'll be very similar in body size. And there was a cool experiment done by an undergraduate at Princeton where she, I think she worked with goldfish, and she dipped one in like a blue ink, and it just stayed temporarily blue, and the predator would target that individual. But if you made all of the individuals blue except for a natural colored individual, they would target the natural colored individual. So it wasn't the blueness, it was just the oddity. Yet that was, you know, that was in the 80s, and there have been relatively few studies since.
Okay, I have just 2 questions. The first one is just to confirm, like, when they are very close, the major force is a repulsive one, and when they are trying to get away, the major force is an attractive one?
Well, it's not necessarily get away. You're trying to move apart.
Okay.
Yeah, it's not like you necessarily turn away. You would move because you're balancing these forces.
Okay. And in this uni-mind perspective, are they able to share some, you know, environmental information to try to rule this movement, like to direction of it?
That's exactly what we'll go on to now. Yeah.
Okay, thanks.
Any further questions? Yep.
In this graph, the size of these circles are also the number of attacks?
Yeah, the number of individuals that were eaten.
Okay.
Yeah, so the color basically represents a model fit. The raw data is shown as the circles. I can't remember what generation this was. We did multiple generations of selection. So again, if you think about these interaction rules, of course this is a schematic. I'll show you that actually these models are only partly right later on. But yes, we have this zone of repulsion, this tendency to avoid other individuals, this zone of attraction, the tendency of a longer range to be attracted to others, and then a tendency in the zone of orientation to align direction of travel. And there may be a blind zone behind the individual where they cannot sense others. That tends to be around 15 degrees in the systems we study, precisely because predation pressure is so important. And so we can think then about individuals self-organizing. There's no one individual that's coordinating the behavior. Each individual is interacting locally, relatively locally, with its neighbors. And by all of the individuals following these same simple local rules, we can approximate the types of patterns that we see in nature, such as these schools. Okay, so I'm going back to this mic. And the first person that sort of speculated this was a guy called Craig Reynolds that I had the luck to meet when I was giving a talk in California once. And he actually won an Academy Award, an Oscar, for his work in thinking about these types of rules. Now, the difference between his model and my model was mine was very, very simple. And I also analyzed it, as I'll show you in a moment. His model was just for computer graphics, so the birds would bank and do all sorts of things. But it was called Boyd's, and it's pretty amazing. It's still, I think, this is the publication here from 1987. But unknown to Craig Reynolds, in fact, in Japan, there had been, prior to this, in the 80s, been a very similar model published. But the thing is, it was pre-Google Scholar. The Japanese work was published in Japanese. And so there was really a sort of independent discovery of these types of interaction rules. But this is, I still think this looks really cool, from 1987, Craig Reynolds showing how these local rules, plus an avoidance behavior of these pillars, for example, could create these types of patterns. And this was used in multiple films like Batman Returns and The Lion King with the wildebeest herds, which is why he won an Oscar. And so these are the two studies that, actually one's from the 70s, that Craig and I didn't know about until much, much more recently. But I think it's important to acknowledge that these ideas had been around, but because, as I said, they were not known until a review paper much later, this kind of was understudied. And so, in a sense, there were these ideas bubbling up, and they just kind of disappeared, or they were used in animation, but they weren't used in biology. And so what I found when I analyzed my model, to actually look at how the parameters interacted with one another, is no matter how much I changed each of the parameters, and I'll come to the details in a moment, I just found that with these three fundamental states of matter. And so if we think about it in our world, in our natural world, if you think about molecular interactions and physical collectives, it's quite remarkable that we have this multitude of different molecular compositions and orientations and so on. And the molecules interacting with each other create macroscopic structures. Yet when we look around us, we see liquid, solid, and gas. So at the collective scale, at the broad scale, there's a sort of hidden simplicity. Right? And so I think the same is occurring here. It doesn't really matter how you play around with these rules. There are other features in the world that mean the dimensionality is reduced. And so, for example, if you change just the speed of the individual, so even if you just keep the rules constant, you do not change the rules at all. The rules are fixed, but you just change the speed, the sort of the energy in the system. So in physics, this would be like heating the system up. But in biology, the individuals are creating their own energy, which is why it's an out of equilibrium system. And here on the y-axis, I'm looking at the angular momentum of the system, the degree of rotation of the system. And what I'm doing in the red is I'm running the model to what's called dynamic equilibrium. I basically run it for an extremely long time, such that the statistical properties are consistent. And then what I do is I slowly increase the speed, or slightly increase the speed of the individuals, and I run the model to dynamic equilibrium. I do the same again and again, and you can see that collective rotation or angular momentum shoots up, and the group spontaneously transitions from the swarm-like state into this milling state, where they're perpetually rotating around an empty core in a random direction. And of course, there was nothing in the equations of motion that said go in a circle. So this is an emergent behavior of these types of interactions. And because there's no gravity in the model, the plane at which this will form is completely arbitrary. Of course, in nature, if you add just an extra force, then it will almost always be flattened out in this direction. If you remove noise from the model, so it becomes deterministic, it flattens out onto a plane. I still don't understand why that is. So there's this inherent tendency to two-dimensionalize the system in that state. If you increase the speed further still, they will transition into this fluid-like group that we typically think of as fish schools or bird flocks. Now, what happens if I do exactly the same experiment, but now instead of increasing and running to dynamic equilibrium and increasing speed, I now decrease the speed. So I'm doing exactly the same as I did before, running it to dynamic equilibrium, and you see now that the system locks into this dynamical state. Right? So you can have two completely different collective states, this milling state and this fluid-like state, for exactly the same parameters. So it's a bistable system. There are two attractors in this particular region of parameter space. And as you can imagine, if I shock the system, I can flip it from one state to another. But if there's not enough noise in the system to flip, you get the behavior like this. So this is called, I'm sure many of you are familiar with this, this is called a hysteresis curve. Right? So it shows that the system itself can have a memory of its previous state, even though no individuals in the group have such a memory or even know what the state is. Now, this is really common in physics, but in biology, people hadn't thought of that before. They thought natural selection just selects on an individual behavior, and that will then result in the evolutionary process. But here we're showing that natural selection can't just select an individual behavior, because you can get different collective behaviors depending on the history, depending on the starting point. And this type of hysteresis and this bistability turns out to be really important in terms of the question that was asked earlier on. What differentiates information processing in collectives versus in the brain of the individuals? So I'll come back to why this is so important in a moment. So in physics, here's a bottle of water that's actually very close to the freezing point, and if you shock the system, just tapping it there, any little particles in the water, bacteria, or can start condensing ice crystals, which can then percolate through the system. So the bottle could be a liquid or a solid state, depending on the starting conditions. Again, a commonly known feature in physical systems, but here we're finding it within a biological system. So let's come to real experimental systems. So it really only wasn't until sort of 2015 or so that it started, or maybe a little earlier, that it started becoming possible. At that time, we had to tag the individuals with these little fluorescent tags. These are made for fish. It doesn't increase mortality for the individuals, but it allowed us to see at least different types of individuals within the group. And these are called golden shiner fish. So when we were doing the study of the predators exerting selection pressure on the prey, I was going out with my lab to Lake Carnegie and Princeton campus, and we were collecting these predators. My lab is mostly physicists, and so they're not necessarily the most competent at field biology. So they were falling over, waders were getting filled up with water. It was a real nightmare to catch these bloody fish. And that was just for the predators. And then I wanted to catch prey fish for these types of studies, and we just couldn't catch them. They were just too fast for us. But then I realized that the fishermen on a bridge actually had buckets full of schooling prey that they were using as live bait, that they were using to bait big predators. And I realized that in the US, they breed these golden shiners for this purpose in the billions. You can get 2,000 delivered to your door for $70, including delivery costs. It's insane. So suddenly I just realized that let's use these individuals, much easier to obtain. And so most of the studies I'm going to talk about are with this opportunistic find, the golden shiner fish. And this is the very first day we put them in the tank and we recorded them. And this is sped up, I think maybe 5 times. The tank is around 2 meters by 1.5 meters. The fish are about 4 or 5 centimeters long. And you can see that they exhibit this polarized state and then transition into the milling state. So I thought, well, this is a perfect system for us to begin to connect and test out theoretical ideas. So at the time, we didn't have as fancy tracking technology as we have now, and you can see the quality of the video is not brilliant. But with Haishan Wu, who's a postdoc in the group, we did a reasonable job with pre-deep learning to maintain individuals for relatively long periods of time. Completely redundant now, of course. And I remind you, these are the 3 fundamental states that we saw, or we predicted from this model from 2002. And over literally hundreds of hours of experiments, we've only ever seen these 3 fundamental states in our experimental system as stable states. And so we call them the swarm, polarized, and milling state. Now, we also saw before that the system would transition just by changing the speed of the individuals. So we asked whether that's the case. So here we're looking at local polarization, so not the polarization of the entire group. If we looked at the polarization of the entire group, here it would be 0 because they cancel out. But if we look at the local polarization, here it would be close to 1, and here it would be close to 1. Okay? So what we see, and the reason that there's 2 curves is there's 2 different ways of calculating this. I don't remember what it was, but I remember there were 2 different ways, and so we show both ways. And irrespective of group size, from 30 fish to 300 fish, as the fish increase their speed, they transition to the polarized state. So basically, when they're moving slowly, they become like a solid, like a dense solid. They're not really moving much, and they're very densely packed. As they start moving, they fluidize, and you get this fluid-like nature, and they could either exist in this or this state. So that prediction that just increasing the speed alone, just adding energy to the system, changes the behavior, seems to hold true. So here we have this low local polarization, and here we have the high local polarization. Any questions? Yeah.
Shows that the number of fish doesn't matter, it only matters the individual speed. But
Yes.
the number affects the individual speed? Like they take longer to...
That is a fantastic question. Yes, it does. So it was surprising to us that it had such little role. I'll come back to the number in a moment. But yes, so it's an autocatalytic system. So for this particular system, the bigger it is, the more sort of energy it has, right? So, but that's not true of every single fish school species that we've studied. But for this one, yes, there was a slight relationship. But one thing that they do is they completely regulate the density. The density, the packing fraction does not change as a function of group size. So it's quite a minimal effect, but there's a slight effect of that.
Just a quick question, because I got lost. This graph, it's a simulation or data?
No, this is experimental data.
Experimental data, okay.
And the measure of local polarization is a sort of average of how smooth like the vector field would be in each of the.
I'll just draw it on the blackboard, but it's basically if you have multiple individuals pointing in different directions, say like this, then you create unit vectors representing each of them, like this. Right, they're meant to be unit vectors. You basically add them together, right? And the outcome will be a vector kind of probably pointing like this, and it will be relatively long. And then the length of this vector here is the polarization, right? So if these, if, for example, they were all exactly pointing this way, you would then be adding 3 unit vectors together, right? And the resulting vector would be over here. You divide it by the number of individuals, and so it would be exactly 1, right? And if they're pointing in different directions, like this, then the resulting vector would actually have a 0 length, right? So that gives you the local polarization, and then we just define it with some range, which I don't exactly remember what it was, but some fixed range around the individual to determine its range of local polarization.
Okay, but what is shown in the graph is the local polarization of one chosen individual.
No, of all of the individuals. The average for all of the individuals in the group, and also for many groups, for that matter. I can't remember how many replicates there were, but there were quite a few.
I think the existence of both the polarized and the milling states is very interesting. And in the model, what characterizes the transition to one or the other?
I'll come to that next, actually. You're right, though. It is interesting.
Yeah.
Okay, so when we look at the time series, again, real data now, when we look at the time series, we see from 30 fish to 300 fish that the system size also changes the dynamics of the transitions between these states and also the probability of being in these different states. So for example, if we look at polarization in blue, the small groups tend to be polarized and rarely have high angular momentum. So small groups tend to be, I can't remember what the groups are, 30 fish, tend to be almost always polarized. A group size of 300, on the other hand, tends to almost always have low polarization and very high angular momentum. So, occasionally, they flip, right? Whereas a group of 70, for example, can sometimes be milling, can sometimes be polarized, can sometimes switch back to milling. And so, of course, there could be multiple factors that affect this. Not least, we have boundary effects within the lab, right? So we work really hard to get rid of boundary effects, especially for group sizes like this, it's quite difficult. But for these smaller group sizes, we can actually exclude boundary effects by filtering the data stringently. And so if we look at 70 fish and we remove the boundary effects, we see that these, if we look at the rotation and the polarization of the system in this sort of phase diagram, you can see these bright spots representing the 3 stable states. The swarm state, the low rotation and low polarization, the milling state, high rotation, low global polarization. Our global polarization here is low, and then high global polarization and low rotation for the polarized state. So you can imagine that the system, this complex biological system, lives in a collective sort of low-dimensional space, right? So the system kind of sits here for a while and flips over here and flips back and then flips there. And the transitional regime, you can see that they kind of move through it quite quickly. It's unstable. So what we wanted to ask is precisely the question here. What's driving the system? You know, this fundamental processes. It could be that individuals change their behavior, and then that changes the collective behavior. Or it could be, that in actual fact there's a metastability, and it's just noise that's flipping the system from one state to another. So just to remind you, that's the swarm, the milling, and the polarized group state. And what we find is that the transition between the swarm state and the milling and the polarized state was really driven by a change of behavior, a change of speed of individuals.
So in the experiment, you're not changing any environmental condition. They are changing the behavior.
In the next experiment we do, but in this one it's just free swimming.
They are changing the behavior.
That's right.
Okay.
Yeah. So there's a natural, you're making really important points because, you know, they are naturally changing speed. Why are they changing speed? We have no idea. You know, it seems to be a contagious process. Once some individuals start moving fast, that encourages others to move fast and so on. But that type of, that has been much less studied. We've looked at contagion of other types of behaviors, but less so that.
Another question, I don't know if we're going to talk about it, but did you test something similar with the locusts? Like, which is the mechanism that make them move together? For example, I don't know if the name in English is lateral line, that they sense.
In the case of the fish, you mean?
Yes.
Yes, so in the fish here, I'll discuss the lateral line actually in tomorrow's lecture.
Okay.
These fish, luckily for me, have a very reduced lateral line. And you can knock the lateral line out with antibiotics. It grows back, the fish aren't harmed, and it doesn't change the behavior. But that is not true of all species. But all of the species we study, they're either very small, and so like in tomorrow's lecture, they're going to be very, very tiny fish, and so they're completely dominated by viscous fluids, and so the lateral line can't work. But here, they have a very primitive lateral line. They're extremely visual. But we also study the lateral line. That's another thing we look into. Are there further questions? And so yes, so remember that the local polarization was high in both of these, but the global is different. Okay, so coming to the question just now, what about if we change the environment? What features of the environment are changing things like the speed of the individuals? How can this be adaptive? You know, when I was suggesting that this allows them to process information, let's look at a specific example. And so we wanted to set our individuals a computational challenge. And so this is actually something very common in computer science. If you're searching a complex data set, you want to find certain types of data or certain clusters of data. Or in evolutionary biology, a fitness landscape, where there's a global optimum, but there are also local suboptimal solutions. Well, these animals face a similar problem. So what I'm going to show you here is an experiment where now instead of just having them swimming in this freely in this big open tank, they're now swimming in a tank. What we're doing is we're projecting onto the bottom of a tank a dappled light environment. So if you go out in nature, they live in streams and lakes, the light's constantly changing. But, and they like to be in the dark regions. And they do so because that's, excuse me, They do so because that's where they feel safe. Now, they feel safe because they're really hard to see. And because they're hard to see, we can't track them. So what you're seeing is exactly the same experiment at the bottom. So it's just a mirror of the same experiment at the top. But in this one, we're cutting out the visible light, and we're filming them under infrared light. Okay, but because we're projecting this time-varying environment, and we're controlling features like the spatial extent of the signal, the degree of noise, the nonlinearity, we changed lots of different things, we can then map that back in. And so this was work with Andrew Badal, who's now professor at University of Washington, and Colin Toney is now professor at Glasgow University. And so you can see them tending to stay in the dark regions. And this allows us to isolate the individuals from this complex background, but put the background back in. So now we can look at both the interactions between the individuals and the interactions with this complex time-varying environment. And what we find is the first experimental evidence that indeed, as group size increases, the capacity for the individuals in the group to find the best regions, the darkest regions, dramatically increases. So there's a real benefit of being in a group. Now, you can see an individual on its own does slightly better. One would be just chance. Does slightly better than chance, just on its own. But being in a much larger group gives a huge advantage in gradient tracking. And so we thought to ourselves, this is terrific. So we now have the data to ask over what range, or how are they responding to their local environment? And how are they responding socially to each other? So the way,
That gives me the opportunity to make a question I was intending to make earlier, but which is, you frequently, when such kind of behavior is described, one talks about collective intelligence, and I have sometimes a little trouble trying to understand what is meant by the emergence of a collective intelligence. And up until now, the processes that appeared seem to be simply the transmission of information in the group. And I was about to ask, the reacting to different stimuli from the environment essentially will still reside in the individual before, because in the model you had simply the way in which one fish will transmit to the other, will influence the other's movement. But in that case, I can see some sort of collective intelligence emerging, because you're showing that the behavior of the collective somehow surpasses what the individual is able to achieve in terms of distinguishing between regions of light and of darkness. If you could comment on
that's exactly what I'm going to do right now.
Okay.
Yep. So I was just going to switch to the blackboard to do so. So what is collective intelligence? So the classic view, and it actually comes from an old study by Sir Francis Galton, who was a cousin of Charles Darwin, and he was a polymath, unfortunately also a eugenicist. But he knew lots of things about everything, but he didn't know much about livestock. And in the late 1800s, he went to a livestock fair where there was a competition to guess the weight of a dressed ox. And so I'm gonna do my amazing picture of an ox here. He shouldn't look so happy because he's just been killed. But Sir Francis Galton realized, he doesn't have much, give it a tail at least, some dignity. So Sir Francis Galton knew that he didn't understand much about this, but he'd realized that almost 700 villagers had already entered the competition. So what he did was he looked at the distribution of their guesses. And the distribution of their guesses was Gaussian-like. And so he simply guessed the mean of the distribution and was 3 pounds off the true weight of the ox. Right? So the classic view of collective intelligence is where each individual in a group makes an independent estimate regarding the solution, and then somehow these estimates are pooled together to give a better overall estimate statistically. You know, of course, it may be that one individual did even better than the average, but on average, this does better. Now, why should this give us collective intelligence? And in fact, when you look at the accuracy of the group decisions as a function of the number of individuals in the group, it tends to go like this. Now, why is that? Well, if you imagine there were only 10 individuals that Sir Francis had, now he'd maybe have a distribution like this, right? As he gets more and more individuals, it's going to go closer and closer to the true distribution. Okay, so this is called the wisdom of crowds. And it's used all the time in human life as well, like prediction markets, for example. You could have a few smart individuals, or you can have tons of individuals and then pool the estimates, or you could somehow do some combination of both. And there's lots of work by my group and other groups because there are many problems with this. Humans also have intrinsic biases. What about if the distribution is not Gaussian, is not unimodal? Then this type of process doesn't work. But in fact, also, what we've also shown is that this relationship between accuracy and the number of individuals only holds if you have Sir Francis's assumptions, which are that everybody makes a completely independent estimate, and everybody uses the same information to do so. Now, if you're in a group together, as you are now, those of you sitting close together have correlated information. Your visual fields are more correlated than they are with someone at the other side of the room. So most of the time, this is not going to be true. If you imagine animals detecting some chemical plume, locally, it's correlated. So if you take that away from Sir Francis Galton's understanding, in actual fact, what you predict is a small optimal group size. And that noise actually benefits. But let's just stick to Sir Francis's explanation for now before we take it apart. So that's how we were thinking when we started this study. We thought that each individual is making a local noisy estimate, independently of the others, of the environmental gradient. And then they're pooling that estimate, not via counting like Sir Francis Galton, but the effective averaging of the social interactions, which means these vectors would somehow be pooled together without requiring explicit counting. In fact, we went even further, and we had a Bayesian model where our individuals were doing this optimally. So we thought we were being very smart. And then we looked at the data. And this is one of the coolest things about doing experiments rather than just doing theory. When we looked at the data, we found that while, of course, the social vector was very important, we found that the environmental vector was completely negligible. What this showed us was that the individuals themselves, irrespective of group size, were not able to estimate the gradient over any length scale or any distance, any temporal scale. There's no temporal integration, there's no spatial integration. The individuals simply do not estimate the gradient.
How can we quantify these two vectors, the social and environmental?
I can't remember. It's too long ago. You can look at the paper, though. Sorry. And so if the fish are not utilizing the local light gradient at the individual level, how on earth can they respond at the collective level? So this is fundamentally different to Galton. Right? Galton assumed that everybody must be trying to solve the problem, explicitly trying to solve the problem, and doing it badly, but then pooling their information together to come to a better estimate when they're as a collective. And this has been speculated, for example, in migration. If migrating birds, if some individuals think we should go that way, some individuals think we should go that way, some individuals think we should go that way, then by pooling this information, as a flock, we could be smarter. But this is something different. And so what we found is that the individuals were just responding to the local scalar measure of the light where they were. So if they're in a good environment, i.e. a dark environment in this experiment, they tend to slow down. If they're in a light environment, an environment they don't like, they tend to speed up. Right? A super simple strategy. It doesn't rely on any temporal or spatial integration, a cognitively minimalist strategy. And yet, the interactions among the individuals give rise to the capacity for the group to detect and respond highly effectively to the gradient. So it's a truly emergent property. But natural selection doesn't give a damn. Natural selection wants to find strategies that work, that are really robust. And so I'll explain to you in two sort of conceptual ways how this works. One is more simple than the other, but the second one is more interesting, I think. So if you imagine my hands are two fish moving together, and there's an elastic band connecting them, or a spring connecting them, and they're moving together on average at the same speed, and then this one reaches a region it wants to stay in, or wants to move more slowly in a nice region. This one doesn't know that, and it continues to move fast. Then what's going to happen to the elastic? It's going to start stretching, and then by stretching, it's going to start exerting a centrifugal force towards the good region. So it's a bit like you know, how a tank can turn, is you've got two treads that can move at different speeds. And if one tread slows down and the other one goes fast, you turn towards the tread that slowed down. Now, it doesn't actually work very, very well just with two individuals. It gets much better with more individuals. But it's just that simple local response with these spring-like interactions that allows the group to do this. Now, this very simple rule has now created a new algorithm in robotics because it turns out to be provably optimal, which we didn't show. This was control theorists at Georgia Tech showed that this is actually provably optimal. It's since been found in a paper in Nature showing that this is how cells climb gradients, because each cell can't integrate across the cell body what the gradient is, but the cells can say whether this is good or bad chemical, or like a high or low degree of chemical. But things actually get more interesting when we start thinking about bigger groups, where the reason that it really starts increasing very steeply for bigger groups is because of what I showed you before. Remember, what happens when individuals slow down? What does the group do when they slow down in terms of the microscopic properties? Yeah, the swarm state or the solid-like state. They're moving slowly and they're densely packed, right? And this is, so they begin to solidify in the regions that they like. But in the regions that they don't like, they're still fluid. And so the system could be tuned to a point of a phase transition whereby you allow the system to fluidize in regions it doesn't like, to solidify in regions it does like, and the outcome is a physical computation at the level of the collective that does not exist at the level of the individuals. It's truly due to the physics of phase transitions and hysteresis. Okay, so that was our hypothesis, that natural selection has found this alternative way. So see how it differs from the classic view. You know, you can't think about this form of intelligence without explicitly thinking of the collective. And the interaction rules only make sense in the context of the collective, which is why this sort of thing had never been studied. And so, well, this sounds a bit group selectionary. I know some of you here said you're working in evolutionary biology. And we should always remember, one of the reasons I study organisms like schooling fish, I do study ants, but I mostly study organisms like schooling fish and flocking birds, because they are of low relatedness. They disperse as sort of plankton. They are very low relatedness within groups. This sounds very much like selection at the level of the group, but it's not. So we can put purely genetically selfish individuals together, and they will readily, natural selection at the individual level, will readily evolve to tune the system, the population even, to be close to the phase transition. And they can do all sorts of other cool computations. They can spontaneously match the size of different peaks in the environment, even though they don't know how many peaks. This is just 2 peaks. We looked at up to 50 peaks of different resource distributions in the environment, and they spontaneously compute accurately how to distribute themselves among those peaks. So it's as if the population is doing a computation, but they're not. It's selection at the level of the individual. So selfish individuals readily evolve to exploit the sudden change of collective state for collective physical computation. And we also have, in this paper, we also took the model, I discussed it yesterday, to the hydrodynamic limit, to the mean field limit, to show that it's genuinely a true phase transition if you deal with infinite population sizes.
What do you mean by peaks?
In this case, we were having gradients in the environment. So like some peaks are just, if you imagine, I'll just quickly switch this over as well. So in the environment before, we had, for example, we had a global optima and also local optima. Well, you can think about it, depends whether you're an evolution biologist, where you think this so this, for example, is the global optima. To simplify the model, what we had was we had different peaks of resources in the environment, right? And those peaks would move around and they could merge with each other. But we also found that as well as finding the best peaks, they would spontaneously distribute themselves optimally among the peaks, even though selection was at the level of the individual, even though they're selfish agents. Yep.
In that perspective of like they speed up when they are not in a good place, they slow down when they are in a better place. I understand these two phases of polarized and swarm, but then when we see the myelin behavior, like I can kind of understand this if like you are in a very good place and then you have a lot of nutrients, so then you, I don't know, try to increase the speed to take more that. But then if you are in a very bad place for like a predation situation, you would speed up and then you would stay in the same place, myelin?
Yeah, you're right. So for example, you can also get selection on the range of the interactions. I remember groups of like 30 or smaller groups would hardly ever show milling behavior. So for example, you may have selection to avoid getting trapped in mills in these types of models. Now, nature is of course more complicated than what any one model. So when you see milling in nature, in many cases, it's when the fish are actually sleeping. So the fish, like these big barracuda or tuna, they need to keep moving because they need to have water flowing over the gills to breathe. They'll drown otherwise. And so they need to stay in the same place, but at a slow motion. And so here, everyone is in the wake of another individual, so energetically it might be efficient. It allows them to stay in the same place. And so the mechanism is probably a similar mechanism, but the selection process is probably very different. And so here, when we have environments with structure, that tends to break the milling down because they're responding to the environment. So these stable mills only tend to form when in very barren environments, typically. So as soon as, for example, in our experiments with the gradients, we never really see milling. You see kind of like milling sometimes in certain regions, but it's always broken down by the environment exerting its influence. And so the milling is kind of partly a pathological state associated with barren environments.
I'm having a bit of trouble understanding what the simulation was in this case. What is meant by readily evolve? You simulated like they did not have the behavior and the populations reproduced and
the populations don't reproduce, the individuals reproduce. So each individual has an evolvable interaction that may or may not depend on, so they can evolve their relationship between the environment and their speed, right? So you can evolve to slow down in good regions and speed up in bad regions. And of course you do that, right? You would do that in the absence even of social interactions because you like to stay in bad regions. But you can also evolve your social interaction. And they would evolve specifically to be in the specific regime where they allow the environment to induce a phase transition, right? So they could evolve not to be in that regime, they could evolve somewhere else, but they specifically evolve to be at the edge of a phase transition at the collective scale. Does that make sense?
Yes, but I'm having trouble understanding how that would be modeled. You simply the population in the initial state, the different individuals have different speeds.
No, it's not different speeds. It's your relationship between how you tune your speed to the environmental features, right? So you can choose. You could choose to move fast through all of the environments. You could choose to move slow. So in this particular model, there was no cost to speed, right? There could also be a cost to moving fast. But here, you just benefit from being in good regions. And so selection, if you're in, say, dark regions, you benefit because you avoid predators. If you're in a food environment, you benefit because you obtain resources. So we abstract that out. So it could be food patches, it could be dark regions, but then there's a benefit to finding or staying in these good regions, and that's all. So individuals that obtained the most benefits, irrespective of their strategy, would be more likely to reproduce and therefore pass on their strategy to offspring. Individuals that had a strategy relating speed to the environment that was very crap, very poor, would not pass on those genes to the environment. And then, because they're evolving in the context of the collective, those individuals that evolved a strategy that when, as a collective, exerted this phase transition-like behavior when they encountered resources, so the group would sort of fluidize and solidify, those individuals would do extremely well. And so groups would form that basically solve the problem via this phase transition. And these, so our golden shiners have inspired little shiner bots, and there's the famous killer bots from Radhika Nagpal, because these cognitively minimalist strategies can then be applied in robotics. You don't need cognition at the level of the individual. You can have these simple local interactions. If they're tuned to allow the physics to solve the problem for you, this can give huge benefits. Okay, so there's genuine practical reasons for doing this type of research. So I always figure it's like a very long period of time to do a lecture, but I don't know whether it's anyone else takes a break for 5 minutes in the middle so people can go to the toilet or whatever. But maybe we could just take a break for 5 minutes and then come back, and I'll show you how all of these models are only partly correct by looking in more detail at the experimental systems. Okay, so let's get back to it. So far I showed you that these types of models are really very influential, very important for us to understand the phenomena. However, the models all make some basic assumptions that come from physics. For example, that individuals interact via pairwise interactions, and that they simply sum them up, so-called superposition of pairwise interactions. And that's a reasonable place to start. But is that actually true within these systems? And also, these models, again, being inspired by chemistry and physics, they had these abstracted forces that individuals were employing with respect to one another. But in fact, individuals are not particles interacting with forces. They are probabilistic decision-making entities that are explicitly basing their decisions on sensory information. And as we discussed earlier, this species almost exclusively uses vision to make their decisions. So one of the other things we did with the advent of tracking technology is to be able to calculate or estimate the positions of the eyes of the individuals and then use ray casting to see how the pathways of photons would fall on the retina of each individual so we could see the world from the perspective of the animals themselves. So instead of considering these abstract forces, we can now consider who do you actually see within these environments. So does the visual representation help us understand and explain these groups better? And so here I've just got a randomly selected individual, this one here, and its left visual field is colored in red, its right visual field is colored in blue. And you can clearly see this is a very complex, time-varying scene that the animal has to make sense of, both from self-generated motion and the relative motion of others. And so what has the brain evolved to do? The brain has evolved, all brains in all animals have evolved to do one basic thing, and that's to take high-dimensional sensory information, complex sensory information about the world, and to reduce that dimensionality, to reduce that complexity down to something understandable in terms of making a decision about the state of the world. It's exactly the same as deep neural networks and other abstracted versions of reality. Now, if we think about the interactions within groups, this has garnered a lot of interest. And there was some very influential work by physicists in Rome who initially looked at snapshots, but then subsequently were even able to track subsections of trajectories of starlings in flight. And one of the cool things that you can see from their paper is that these groups look to us to be very volumetric, but that's actually an illusion. It's called the Moire illusion. You know, if you have a relatively two-dimensional object, like a comb, for example, and you move the comb with respect to light, you see waves propagating across it, waves of density propagating across it. And if you actually rotate the tracks of the positions of the starlings, this is just two particular trajectories, they're basically flying as a sheet. It's a sort of two-dimensional system that looks three-dimensional to us. And they also found evidence that it depends on topological distance rather than metric distance. What is topological distance? Well, metric distance is distance in space. Topological distance is the number of individuals you interact with irrespective of space. So, for example, I could interact with 4 nearest neighbors irrespective of how far away they are from me. So it becomes a scale-free form. And so they find evidence, indirect evidence, I hasten to add, that topological interactions were more plausible than metric interactions for their system. But what we can do in our experiments is we can actually look at the time-varying dynamics of the system, and even more importantly, we can conduct perturbations to the system. So we can explicitly test between different theories of the interactions. And in our experiments, we were able to ask whether the interactions, whether the change of velocities and the change of direction of individuals, the way it propagated through the group, was best explained by metric interactions, topological interactions. What's also been proposed is that there are Voronoi interactions, that you just interact with the first and second layer of your Voronoi neighborhood. And of course, we added a fourth one, which is the possibility that they interact with visual interactions. And all of the models had the same number of parameters. And so we could then use information theory or Bayesian model fitting to work out which of these theories best fits the experimental data.
Yeah. Could you explain again the difference between the metric distance and the topological?
Sure. I'll just switch over to the blackboard. So metric distance, the one parameter in the model that you're changing is basically this radius here. Right? That's the only parameter we changed, right? Your metric distance might be large or it might be small. And then you're just assumed to interact with all individuals within that neighborhood. In the topological model, you interact with a fixed number of neighbors, say 3 neighbors in this example, so you would not interact with this one, but you'd interact with these 3, irrespective of their distance. So a single parameter, again, which is the number of neighbors you interact with. So we can look at 1, 2, 3, 4, 5, 6. Here we can look at different distances.
You mean irrespective of their distance, because they may be very far, but you choose the n closest to the individual?
Yes,
okay.
That's the assumption. You choose your n closest near neighbors. And then the Voronoi doesn't have parameters because it's just the Voronoi neighborhood. And then the visual model has a single parameter, is that you interact with all individuals that subtend a larger than specific angle on the retina. So individuals that subtend only a very small amount would not impact you. And again, this is a continuous term, and the model fitting then compares all of these models for all of the parameter ranges. Right? So each is a continuum. Here a continuum of R, here a continuum of n neighbors, here a continuum of omega angles. Right? So it's a fair comparison.
And in the case of the visual one, you will tend to interact more with the ones whose directions are different from yours. Okay.
Exactly. So I'll come to that later because I thought that would be important, but it's not. But it's a really good point. But we'll come back to the geometry shortly.
Professor, and the Voronoi is just a control? The Voronoi?
The Voronoi is because it was proposed by a group in Toulouse, and we thought we should incorporate it because it turns out to be useful. Yeah. So it's not really a control. It's a you'll see when I show the results.
Okay, sorry.
And so what we find is, now note this is a nonlinear axis. So the visual model here fits much, much better than the metric and topological. The metric and topological do better than all-to-all interactions, so there's definitely something local about the interactions. But note, the Voronoi does quite well. If I was running a model and I wanted to sort of approximate the interactions in a cheap way, because calculating visual fields is a pain in the arse, Voronoi is better than topological and metric. Right? So the Voronoi model actually captures a lot of what you need, but you can see the visual still, nonetheless, does much, much better.
Sorry, how can we construct the polygons in the Voronoi?
Sorry, I couldn't hear you.
How can we construct or build the polygons in the Voronoi metric?
It's pretty much standard in most software packages to calculate. You can calculate the Dirichlet neighborhood or the Voronoi neighborhoods.
Okay.
Yeah. It's just all of the points that are closer to you than anyone else is within your neighborhood, and there are very fast algorithms for calculating this because it's so commonly used.
Okay.
Yeah. It just kind of represents your first shell of neighbors, so to speak.
Sorry?
It sort of represents your first shell of neighbors around you.
Okay.
We also discovered that it's a fractional contagion process, and that's going to be important for tomorrow's lecture. So basically, you're looking, so if individuals change state, like say suddenly individuals startle, or individuals suddenly move fast, or move in a directed way, it's not just the number of individuals that do that, it's also the number of individuals that don't do that. And specifically the fraction of individuals that do one thing versus another. And that turns out to be ubiquitous, and we think we understand why. But that's not so important for what I want to say right now. That will come in more tomorrow. So we can then ask, well, we can then construct networks based upon the best fit for each of the models. And we can then look at network properties. This was a long time ago, so I hope I remember this. But the efficiency was how quickly, you know, if you're passing, say, a state or information from one part of the network to another, how many hops does it need to make to get through the system? Like to get from any one individual to anyone else. So having a high efficiency means you need less hops to get the information, less connections, less paths to get the information through. And you can see that the visual model, the red, requires slightly less hops. So the information flows through slightly faster than the other networks, but it's only slightly faster. However, if we look at the transitivity, you can see there's now a dramatic difference. And the Voronoi, interestingly, also has a very low transitivity. So what is the transitivity? I think I added in the definition because I haven't talked about this for a long time. The network transitivity is the extent to which individuals who share a neighbor are neighbors themselves. Higher transitivity indicates a greater likelihood of one's neighbors being mutually connected and hence a greater level of redundant information. Right? And if we're thinking about collective intelligence, if you have correlated information and you're accumulating information that's correlated, that removes the potential for collective intelligence. You want to decorrelate information among your neighbors to maximize decision-making effectiveness. And it so happens that these visual networks, because the visual networks, of course, come from the geometry of the system. The geometry of the system is under selection pressure. And one of the reasons this might be is that these networks have extremely good characteristics for conveying information of any type. And when I talk about information here, we'll get into that more tomorrow. It can be, for example, a predator suddenly appears and individuals will turn away, and that information needs to propagate very rapidly through the system. Or that individuals speed up to go towards a food source. So any of these types of information will tend to flow through the system much more effectively. And again, the metric and topological neighborhoods do particularly badly. So I think it's unlikely, in my opinion, that topological neighborhoods are ever really used in nature. It's much more likely that they're using visual information, and it just looks like topological information because the visual information also has that scale-free property. Unless individuals move really far away, you can still see them and still interact with them. So I think I'll leave it there today, but tomorrow we'll go into more details about social contagion. So this tendency for individuals to change state within a group and percolate through the system. And in particular, we will compare our understanding of epidemiology. So people used to believe, until this study that I'll talk about, that behavior basically spreads like a fast-spreading disease through the network. And it's intuitive that it should spread in such a way, but it had never ever been tested. Almost all the models just assumed that it was true. So tomorrow I'll show you that it's fundamentally not true. And in fact, the network properties that maximize the spread of social contagion are those that minimize the spread of disease, and vice versa. And so the strong selection pressure that disease has had, or the potential for disease spread has had on these groups, is also likely important. But I think it's too much to get into now, so let's do that again tomorrow. Cool, so let's end there for today, and then we can, I think it's too big a section to get into right now. But I'll take, if there's any further questions though, a good time to ask.
This slide, the degree that you're showing is the degree in relation to the individual in the, yes, this one. This or the other one, the x-axis.
The degree here is the number of neighbors that individuals are connected to, so the degree in the sense in the network degree. And I should have pointed out that the open circle, square, and triangle, that's the best fit, right? So that's where we think the system actually lies, but because we can't be absolutely sure, we also looked around those points to show that the results here are not dependent on the exact degree distribution of the network.
in the topological one, these things are fixed because you fix them.
What are fixed?
The number of individuals. That
Yeah, we look at across the whole range of the number of individuals that individuals could interact with, yeah.
That's why in the visual it stops at 1, and in the Voronoi it has only 1 parameter. In the visual you don't have until the end, because you have a limitation of...
Yeah, because in the visual model, it's, you can't interact with individuals you can't see,
Okay, yes.
right? Whereas in the topological model and the metric model, in principle, you could interact with everybody,
Yes.
right? The visual model is the only model that preserves the fact that there's blocking of information from others. So that's why the visual model can only extend to a certain degree, unlike the other models, which is another, obviously, realistic feature. If you can't see individuals, especially for longish periods of time, it's unlikely when they respond, you can respond to that directly, right? So it's kind of amazing to me that this, 2013 was the first time anyone sort of thought, well, you know what, it could be visual, because it's just such an obvious thing to think about. But what was perhaps more interesting was the fact that these visual networks they have a very different characteristic than the other networks. They're sort of more abstracted networks. Except, as I keep saying, Voronoi does a pretty good job. If I wanted to play around with a model, an abstract model that didn't have complexity, I would choose Voronoi interactions because I think it comes the closest to the real nature of the interactions in the animal groups.
Okay, thank you.
And of course, the Voronoi also has a scale independence, right? It doesn't matter how far your neighbors are away from you, you will still have Voronoi neighbors. So it's a bit topological-like in some respects.
You only have 1 point in the Voronoi.
Yes, the Voronoi, you've only got 1 shell of neighbors.
Okay.
So that's, you could potentially extend it and say, well, you interact with 1 shell and then maybe a little bit of the next shell or something like that. You might be able to approximate the visual model more accurately with the Voronoi model, but just taking Voronoi out of the box, just looking at your Voronoi neighbors, does a pretty good job. And so Guy Terrelatz and others in Toulouse who use the Voronoi model, I think it's a reasonable approximation for most purposes.
It's, for me, very interesting. This, I mean, I guess I can accept it, but it's initially, for me, it's interesting to see that the Voronoi and the visual are the ones that are doing better, because a visual interaction is very long range, right? It can be very long range, whereas the Voronoi is extremely local.
It can be long range, depending on how you tune, or how the system tunes the angle, right? Mm-hmm. But you can be long range. And I think the great thing, it is surprising to me also, the Voronoi, but it doesn't, I mean, it does well, I think, because it captures these network characteristics. But it doesn't do very well. I mean, there's a big difference between the Voronoi and the visual in terms of the actual fitting. But the Voronoi, I think, does quite well in capturing the types of network characteristics that are most important. But it doesn't do nearly as good a job at predicting how information flows because, exactly as you say, it misses these long-range possibilities. And, and again, it shouldn't be that surprising. Because of the fractional contagion, it doesn't matter if what's happening is far away. If you see individuals changing state, like suddenly fleeing, you want to be able to respond to that. You don't want to restrict your neighborhood. So it makes sense to have, but we also looked at the structure of these interactions in much larger groups, not in this paper. And they're not small world or scale-free networks. The visual networks really are in a sort of unique class of network structures as well. So it's not clear to me I think that much more work needs to be done. And also, as I'll show next, these networks here are simplified versions of what we'll get into next, because in actual fact, as was pointed out, the geometry is really important, right? An individual, in this orientation in front of you is going to extend a big angle in your retina. The same individual over here will be a small angle, and it will be a big angle again. So there's all these other features that come into play that actually change the weighting and the directedness of these networks. So it turns out that these are highly directed networks. Like, I might be strongly influenced by you, but you not by me. And so, again, we're kind of building our understanding from 2013 up a little bit. So we'll get into more realistic models and models that are capable of making specific predictions about who will be infected by behavior, when and how far it will spread. So we get very, very precise in the next lecture about this. So but this is like a sort of our stepping stone to say, look, vision really is important. And next what we'll do is we'll look at the visual information that they get and work out which pieces of visual information are important. And then construct these weighted directed networks and then show only by adding that component can now this stochastic system become highly predictable, extremely predictable.
Yeah, I think one thing that I sort of would argue to convince myself that the Voronoi should be somewhat good compared to the approximate the visual is that your Voronoi neighbors are kind of the first occluders of your vision, right? Is what you were pointing towards.
Yeah, exactly.
So
and
you're missing some things that leak long range, but
exactly.
they are the first occluders of your.
Exactly right.
And
I think, you know, the Voronoi neighbors probably do a good job in certain parts of the group, better than in other parts of the group. You know, so I think, so this is, as I said, this is like a stepping stone. This was the first paper that said, you know what, it's because everyone's just obsessed with metric and topological. All of this, hundreds of papers, metric and topological. And it's like, let's slow down here and actually look what it is. And this sort of suggested, you know, visual does better. Very simple visual does better than metric and topological. But we can do much better than that still if we take into account, well, vision is a lot of possibilities, right? There's many things you could pay attention to. And so we end up figuring out what those are as well.
I feel a bit lost because I feel like we abstracted a bit from the system. When you compare this to what is expected, it is what is expected to a specific kind of fish and its collective behavior, or because, for example, you mentioned that the bird flocks, they tend to be two-dimensional surfaces which are changing, whilst as far as I could understand, the fish are still seen as three-dimensional, as volumetric.
No, this is all done in quasi 2D. This is all done in 2 dimensions. And so the fish that we study, they swim largely in 2 dimensions as well.
Okay.
Yeah.
And is there a reason to think that perhaps such a decision that we're trying to make between the different kinds of local interactions will vary between different kinds of fish? Because
well, some fish will not use vision. They will use predominantly the lateral line if they're schooling in the dark, for example. So that could produce a fundamentally different structure. Okay. But I think, I would guess for most species, almost all species, this would be appropriate.
Very nice.
Yeah. But as I said, tomorrow we'll discuss in more detail about the actual sensory modalities they use. And then we'll introduce virtual reality and other things that will allow us to warp space and time in ways that are not warped in nature. So we can test. So I'm going to build, you know, this is 2013, so it's now seems outdated, but by the time we get to 2022, you'll see that we've made a lot of progress. But I didn't want to jump to the end. Well, it does get quite complex, like, because, you know, it turns out that they don't respond to each other in Euclidean space, for example. Everything so far has been So everything so far has been in Euclidean space, right? Everything so far has been in normal space-time. But it turns out the brain warps space and time. And the only way we can explain their decisions is that the brain does so. And the only way we could study that the brain does so is by creating immersive virtual reality environments where we can play around with space and time in ways that you can't in conventional experiments. So it gets pretty crazy later on.
I'm very excited.
That probably won't be the next lecture, but the lecture after. I guess are there 2 tomorrow? Yeah, I think so. So we'll probably get into it. We'll probably get into it tomorrow. So tomorrow will all be about geometry. So I'll get through the story about disease transmission being very different to social transmission, and then the rest of it will really be about how animals represent space and time. Cool.